is probably, Sean probably mentioned, you have a palladium catalyst or platinum catalyst.
Yeah.
With triglycerides, it's often nickel, high heat, but the platinum kind of has those hydrogens dangling up, up into space, and so we then hydrogenate these triglycerides doing that. So we break the double bonds, tack on hydrogens, and this is how we make margarine. Good old. Anybody have Country Crock when they were a kid? Do you guys remember this was all the rage when I was a kid? Yeah, Country Crock. So it's funny that it's a crock because margarine kind of is a crock. But it was touted as the healthy alternative to butter because it wasn't saturated fat. And as you guys all know, nutritional advice changes. Right? But the idea was that they would take soybean oil or peanut oil and partially hydrogenate it. So they didn't completely hydrogenate it. They would hydrogenate it to a certain extent so that it had a solidifying property to it. Okay? But the problem is that in some cases, when you're doing this hydrogenation step, you can get these fatty acids to isomerize, and instead of having that cis, like I kind of pointed out right before break, you don't have cis, you can get trans.
That's from the hydrogenation stuff.
Yeah. And we ate trans fat for years thinking that it was a healthy alternative. The problem is that if our body has adapted to processing cis fatty acids, then it may not know how to process trans. Oops. And that's exactly what we see. Okay, so oftentimes these trans fats, they get broken down from their triglyceride into their fatty acids, or maybe they stay as triglycerides, get stored in your adipose tissue, and then are never hydrolyzed again. And so you have these molecules building up in your body that can't get processed. And so it's been associated with cardiovascular disease. The whole clogging of your arteries. And if they become oxidized, now they're toxic. And so those trans fats, it's conceivable that they might be in us forever. What makes fat cells increase is the number of triglycerides that are put in there, right? As we put on more fat, it's our cells getting bigger. It's not our cells dividing more, right? Adipose cells swell up when they're full of triglycerides. If you start needing to use those triglycerides, or you're not consuming a lot of triglycerides, they shrink down. Okay, we start hydrolyzing them, we start using them to make acetyl-CoA to put into the citric acid cycle to make ATP, right? Well, if you are consuming fats, triglycerides that the body can't break down into acetyl-CoA, then they just stay in there. And so, if you had a lot of trans fats, it's conceivable that you would never be able to shrink those cells because they would not be broken down. So the FDA phased out trans fats in 2018, I think. Not that long ago. But I don't think, you won't find Country Crock with trans fats. You may not find Country Crock anymore at all. been looking for it, but in the 80s and the 90s, margarine was the healthy choice, and then it went back to butter, and I don't imagine it'll go back to margarine, but you know, who knows, things go back and forth a fair bit. All right, so the other thing, auto-oxidation. So when we have oxygen present, which is going to be in most cases, right? That bottle of oil, you've opened it up, right? Oxygen then goes into the bottle. A lot of bottles are dark though, you'll notice. Olive oil usually comes in a green bottle, not a clear bottle, right? And that is to reduce the UV light from coming in and creating oxygen-free radicals. Beer, same kind of thing. If beer is in a brown bottle, it's just going to taste better than if it's in a green bottle, like Heineken.
Yeah, I guess it's true.
And if it's in a green bottle, like Heineken, it's going to taste a little bit better than if it's in a clear bottle, like Corona. Right? Brown limits more UV than green, and green limits more UV than clear. Right? And so, if we have an unsaturated fatty acid, it's going to be particularly susceptible to auto-oxidation. Okay? And it's going to happen at the allylic position. Right? The allylic position is going to be the side that's closest to that carbonyl. And it's going to create, as a result, this hydroperoxide. And the steps for this, and you guys did some degree of radical mechanisms with bromine and stuff like that, and the peroxidase, or the epoxies and stuff like that, right? It's going to be the sum of the propagation steps. So you guys did initiation, propagation, termination, right? That's all coming back. Okay. But essentially, we can cross out things like we do with net ionic equations, right? And get our simplified net reaction of whatever our hydrocarbon is, in the presence of oxygen, it's going to make this hydroperoxide. Okay? And so this is what's happening in those bottles of oil. Okay? This is why you get that rancid, and they call it's not my word, it's called, oil is termed, it goes rancid. It can have a really bad taste or a really bad smell to it. Nuts. Oftentimes they'll say keep certain nuts in the refrigerator or the freezer because the nuts have oils, and those oils are polyunsaturated, and if they get oxidized, they get rancid, and it's because of this process here. Okay. And then now you have a peroxide that you consume. What can happen? That peroxide can then reverse, put the hydrogen back on, and now you have a free radical in your body. What's that free radical going to do? It's going to attack the next thing, which happens to be your biomolecules. And as you folks all know, when you change a molecule in a very simple way, the structure, you change the function, right? And so toxicity can result by altering the structures of our biomolecules, the things that make us us, by altering enzymes or altering proteins, altering signaling molecules, right? Cancer, right? All of these things. Now, just like I don't want you to go home and fear prions, I don't want you to go home and fear oil, okay? But this is an explanation of that kind of thing that can happen. You take triglycerides and you
put them in the presence of a strong base like sodium hydroxide, you will cleave off the glycerol and separate the fatty acid. So biosynthetically, we do this with lipases, okay, so that we can absorb these. But if you want to do this in a lab or at home, you can take oil or any triglyceride, and you can treat it with a base. And interestingly, this is how we make soap.
Okay.
Saponification is essentially making soap. Anybody watch Fight Club?
Yeah.
They go to the liposuction clinic. They get a bunch of fat and they render it, and they have that scene. Have you seen Fight Club? Okay, well, I'll mention it. You've seen Fight Club? Okay. There's a scene where he kisses his hand and he pours lye on it, and it burns, right? Lye is super basic. The lye is used to break apart the fatty acids from glycerol. Okay. So that they can make soap. But they weren't making soap just to make soap.
Making bombs, yeah.
So, gotta be careful, Siri's probably listening to me. But you can take this glycerol, right, as a byproduct, and turn it into nitroglycerin. Okay, so you essentially use fuming nitric acid, so super concentrated nitric acid, and it will tag on these nitrate groups. Do we talk about TNT? We talked about TNT in here, right? Trinitrotoluene. Those nitro groups are highly unstable. So if you put it on a benzene ring, kablooey. If you put it on a fatty acid, you convert a glycerol. So here's the 1, 2, 3. Right, we've removed the hydrogens and we've tacked on those nitro groups. It makes it very unstable. And that's when you get nitroglycerin. Okay. Now, interestingly, nitroglycerin can be an explosive, but it can also be a medication.
Really?
Yeah. So people take nitroglycerin because it's a vasodilator. And so folks with heart conditions like angina will, and there might be different drugs now, but for a long time, I'm talking over 100 years, this was used as a medication to dilate the vasculature. And what this creates, the reason this creates soap, is because you have this sodium ion that is attracted to this polar group here, and that makes this head, this is the head, polar, and then this non-polar hydrophobic tail. So what soap does, right, it binds to two different aspects of this spectrum, okay? So if you have bacteria on your hands, bacteria are cells. Their cells are made of, we haven't got to it yet, but phospholipids, right? So the non-polar tail, like dissolves like, right? It binds to the phospholipid part of the cell. And then the polar head has an affinity to the water that you're washing your hands with, and so you're washing away the bacteria. And soap makes those bubbles because it's trying to, the polar part is trying to get away from the water. And a soap bubble is round because that's the most energy efficient distribution to get away from something, right? So that all of them can be as furthest away as possible. It's a sphere. Okay, and on the inside of that bubble is going to be your non-polar tail, and it's going to trap in anything. Right? So, any kind of oils, right? You cook, you put some olive oil in there because it's got a higher smoke rate than, say, vegetable oil. Vegetable oil is very polyunsaturated, and so it's going to oxidize more quickly. You cook, what are we cooking? What are we doing?
I don't know. Eggs and eggs, I guess.
I was going to say eggs. We're on the same brain. Alright, we're making some eggs, and we're done, right? Do we just take that and put it under the water in the sink and then dry it? No. We gotta use soap, right? Because the water itself is polar, the oil is non-polar, so they're just going to splash off of each other. The soap bridges those gaps, right? The soap, non-polar part binds to the oil, and then when you put water in it, this polar part binds to the water and takes it off the pan. In our body, fats and waters don't mix, right? But we can have a way to get around that by using bile acid. So your pancreas makes bile. You've all probably experienced bile if you're dry heaving. It's that stuff when everything else is gone, that yellow, nasty stuff, right? That's bile. What is that for? Why is that inside? Well, the bile is like an emulsifying agent.
Okay.
An emulsifying agent, if you don't know, is something that, kind of like soap, can bring polar and non-polar things together so they don't separate out. If you were to make your own salad dressing,
Yeah, Caesar salad dressing is just
Right.
saying, yeah.
you use oil, and maybe you use vinaigrette of some sort, or if it's Caesar, you use, hold on, don't tell me, you use the little sardines, right?
Yeah, anchovies.
The
Yeah.
anchovies, that's it, yeah. But if you were just making a vinaigrette of some sort, you could put a little bit of mustard in there. And mustard is an emulsifier. It has both polar and non-polar parts, and so it keeps it from separating out.
Once it separates, it's hard to put it back together.
Yeah, and all kinds of sauces and dips and things have these emulsifying agents in them, so that when you buy it from the grocery, it's all nice and consistent. You don't have to shake it up much. But the bile acid does the same thing. It allows the triglycerides to essentially emulsify with that bile acid, so that it's not repelled by our mostly polar milieu of our body. Questions on that? Mechanism. So how do we do this? How do we hydrolyze? We have our strong base, and it's essentially 3 steps: nucleophilic attack, loss of a leaving group, proton transfer. Okay. The strong base is going to attack that carbonyl carbon, and we're going to break up that carbonyl, and then we're going to get a loss of a leaving group to give us this intermediate here. We can then do a proton transfer and end up essentially with an alcohol here and a carboxylate ion. And that alcohol is, if it's a triglyceride, it's going to be the glycerol molecule, right? So there'll be 2 other alcohol groups on there. But if we're looking at just in isolation, that's the process that's happening.
You can do this under acidic conditions too, I believe, but basic is much more common. And they think that soap was kind of just serendipitously discovered when people would go down to, say, a river and wash their clothes, because it's water, and they realized that water is good at that. And now it's a beaver or something dies, and it's in the water. Beaver has fat, and sometimes, you know, if there was just the right basic conditions, that could create a little bit of soap in that river. The other thought is that, well, maybe people used to bury their dead near rivers, and they would throw lye on them to keep the smell down and help with decomposition. Right? And if it was close enough to a body of water, the fats in the deceased bodies would mix with the lye and then leach into the water, and all of a sudden they see these suds. And someone has it, I'm gonna stick my clothes in there, I don't know, but somehow they made that leap that you know, let's try it.
Yeah. Clothes are cleaner when you wash by the graveyard.
Yeah. And you know, they used to make fat. You can make fat, you know, around the campfire. You just take fat from an animal and mix some potash. You can use just literally the ashes from your fire are going to have basic characteristics to it. Mix in some of that campfire ash with the fat that you've taken off of this animal and render it into a soapy consistency and get saponification that way. Okay, what is this? Identify the products expected when following triglyceride is hydrolyzed. All right, so essentially, what are we going to do? We're going to break the bonds here. So what are you going to be left? Write that out. It's a little bit laborious because you got to count all these carbons. But what would you get when you hydrolyze that triglyceride into fatty acids? We get it? We still working on it? So we'll use excess
sodium hydroxide, something basic, and we'll end up with our glycerol. And then we'll have our carboxylate, so and then the sodium's going to be hanging around there. And then however many carbons, right? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15. And then same thing here. I'm going to put an R because I'm lazy. And then this last one, you know, same thing. Only you got to draw out this interesting kink here. But notice it's polyunsaturated, but they're both cis, right? Natural order of things. No trans fats typically exist in common matrix from that artificial hydrogenization. Right.
So do plants use fats for the same thing? Do they like store it for energy, or do they store more energy as carbohydrates? Or?
Yeah, no, it's both, right? So plants are, plants also have a lot of wax, right, for protection. Their cell walls, they have a cell wall, unlike our membrane, so it's a little bit different in structure. But so those oils the oils can have different purposes, and I'm not a botanist, so Sue Kloss might be the better one to describe this, but I imagine that oils are typically going to be most concentrated in things like seeds, where they need that energy for the germination process, right? So if you think about a leaf, like think about all of the oils that you use. Do you use spinach oil?
Like grapeseed oil?
No, right? You use oils from the seeds because that seed has to have a bunch of nutrients to put into that initial germination and that what do they call it? The seedling or something, you know? I'm terrible on the plant side, but it's an energy source.
Okay. Yeah, it's just interesting because we keep like adipose tissue all over, but plants don't seem to have like fatty tissues like that.
No, and also interestingly, if you look at a lizard, ever see much fat on a lizard? Or a snake? That's a fat snake. You don't see that, right? They are cold-blooded too. They are what's called poikilothermic ectotherms, if you want to use a nerdy way to describe something as cold-blooded. Warm-blooded is homeothermic endotherms. So homeo stays the same. Endotherm, we get our energy, our temperature from within. Poikilo means to fluctuate. Ectotherm, they get their temperature from the sun, right? From the ambient heat that's out there. The reason in one aspect is that they don't have a lot of fat. And they don't have any brown fat. And the brown fat is what is particularly used to maintain body temperature. Same with fish. I mean, fish have some fats in them, and they have the omega-3 and omega-6 fatty acids. But they don't tend to accumulate it in the same. And if we're talking about accumulating adipose, humans are really the only ones that have it, unless we have domesticated animals that we then put that on. But nature, most organisms don't have excess of that. But yes, the distribution in animals is to have it kind of throughout. You got to move, right? We have muscles. Those muscles need ATP. A plant doesn't do a lot of movement, right? They grow, but that's through mostly cellular division. And so they don't need to have the ATP at hand for locomotion.
That makes sense. But for the seeds, though, it's different. Yeah.
But the seed needs that energy to put into all of the signaling cascades that, like, so plants have all of that too, right? They need to send messages based on the external environment. They need to have enzymes to undergo photosynthesis, right? And so, and they are also not just photosynthetic. They use the citric acid cycle as well. They don't just make all of their sugar; they also can do cellular respiration.
Okay.
Which I didn't know. Somehow I missed that in college. I didn't realize that until I was teaching Bio 110 once. It's like, oh wow, yeah, they're not just photosynthetic. They can, of course, make sugar. We can't, but they can also use oxygen and sugar to make ATP.
Yeah, we'll talk about that in Gen Bio.
Yeah, so they can do both. And it makes sense because, getting off topic, but whatever, it's the last couple lectures, we can get off topic. It's believed that there's this thing called endosymbiosis, where two single-celled organisms came together, right? Essentially one ate, the big one ate the little one, and the little one didn't die. The little one just stayed inside, right? And so if you are a cyanobacteria, which is a single-celled organism that is photosynthetic, and you were eaten by a bigger, single-celled organism and didn't die, then now you've been co-opted and you turn into chlorophyll, the things that make photosynthesis from the plant. Same thing with mitochondria, because mitochondria has its own DNA. You get it from the mother, and mitochondria was probably at one point its own, it wasn't an organelle, it was an organism that got internalized into another organism and then co-opted to just be a powerhouse. And it's symbiosis because now that organism has a home, right? It's protected. Yeah, it's been co-opted to just churn out ATP, but it's got protection, right? Same thing with the cyanobacteria. It's protected, and now it can make sugar for that organism. So plants, right? Plants and animals had a common ancestor at some point, right? And that common ancestor was probably doing cellular respiration. And then where they diverged was when one organism internalized something photosynthetic.
To make the chlorophyll.
But it still has its ability to do cellular respiration as well. And if you put plants in, I know I looked in the biolab once and I don't know if it was Bio 102 or if it was a botany class, but they had a bunch of cut flowers in different treatments. One was bleach, figure out how that would work. But there was just water, DI water, and sugar water. And the one that was in sugar water the flowers lasted longer.
Oh, right.
And you get those packets when you buy flowers. I imagine there's probably, I don't know what is all in there, but I imagine there's some sugar in there. Because in addition to making sugar via photosynthesis, they can also take in sugar and use that as well.
Okay. That's interesting. I didn't know that.
What do we got? Okay, yeah, let's wrap it up with triglycerides, and then on Thursday we'll go into phospholipids. So, triglycerides, very good way to store energy, right? We can pack it away into cells, they'll stay there until we need them. They have more kilocalories per gram than other biopolymers. They generate more ATP, right? So all in all, they're good. So it shouldn't be too much of a stretch that we can use that in diesel engines. And in fact, in World War I and World War II, they would use this process of transesterification to make Well, I don't know if they did this actually. They might have just used straight up coconut oil, which you can't. It's not going to be as efficient though. You can convert it to biodiesel, okay, by essentially making breaking off the glycol and putting on an ester group. And that's how you make biodiesel. You may know biodiesel, some people go around, they get the oil from fast food restaurant, like the used spent oil, and they use that to convert it to biodiesel.
I worked in Truckee and we had like a receptacle for our oil, and then the biodiesel company would come and take that to make
Yeah.
biodiesel.
And
with
then
it.
they do this.
Yeah.
But you can burn straight up vegetable oil. Like you can use it to combust and use it. It's not very efficient. You get a bunch of carbon soot build. It doesn't burn cleanly at all. But if you esterify it,
Okay.
then you get a better product. It's more efficient. And when I was in grad school, I came across a paper. I don't even know why, because it wasn't related to anything I was doing. But some group was making biodiesel from coffee grounds and found that it was way more efficient in the yield. So you could get upwards of 90% biodiesel from the coffee grounds, as opposed to vegetable. Like the vegetable oil from the deep fryers and stuff, it's doesn't approach 90%.
Yeah.
It's like 70-ish or something. But I was like, maybe that'll be a fun garage project, because I drink a lot of coffee.
Yeah.
And rather than just throw out the grounds, maybe I'll just make biodiesel. I'm like, what am I going to do with biodiesel? Like, I can't drink that much coffee
Yeah.
to convert my car to biodiesel. So maybe I'll convert my lawnmower. Maybe that'd be enough. And then we had our first kid, and so all of those silly things went away. But yes, anything that has oil can be converted into biodiesel.
Maybe for your camper, there's little diesel heaters. You could use
Yeah,
for that.
right. Maybe, yeah. Well, if I ever get back, maybe when the kids get a little bit older, I'll revisit that.
Yeah.
But
Cool project.
I wonder if coffee houses are going to start giving their grounds to biodiesel companies.
Oh, really?
Like restaurants do, maybe.
maybe. Oh, I thought you said yeah. I mean, that would be.
But maybe.
Yeah,
Yeah,
good way
that'd
to
be nice.
reuse
I mean,
it.
you can use the grounds as compost and
Yeah.
fertilizer and stuff. It has a lot of nitrates and stuff, but or not nitrates, but nitrogen. But yeah, a coffee bean is not actually a bean, right? It's a berry. It's the fruit. Naturally, it's going to be oily, right? When you guys all drink? Are you drinking coffee? Okay. You drink coffee?
Oh yeah, I love coffee.
Nikki? No? Oh.
Yeah.
You haven't watched Fight Club. Don't drink coffee. No. I used to be inherently suspicious of people who didn't drink coffee. It's like, what? But now I'm actually, I admire people who aren't slaves to caffeine. And we could talk about caffeine. That's a really interesting thing, the way that works. But we don't have time for that. Maybe some other time. But maybe next week, because caffeine mimics adenosine, which adenosine makes up our nucleotide, one of our nucleotides, and also makes up ATP, right? So yeah, remind me, I'll talk about why we shouldn't be drinking coffee, because it just ruins your baseline. You remind me, and then I won't feel so bad about making you feel alienated for not watching Fight Club and drinking coffee. I talked to a guy who used to roast, and he would say that the really oily beans, if you open up a bag, some of them are just shiny, right?
Yeah.
You open up another bag, they're not. They're really dry looking, right? And I always thought the dried ones were over roasted, like the oils were just broke. They roasted the hell out of that. There's no oils left, right?
Yeah.
He told me it's the opposite. If you over roast it, then it forces the oils out of the bean.
Okay.
And those oils have a lot of flavonoids and polyphenols and antioxidants, right? which, I mean, they're still on the outside, but now they're being oxidized, right?
Yeah.
So, a really, truly well-roasted bean has a dry consistency. Doesn't mean the oil is all gone, it just means it's still inside the bean.
Okay.
And that makes total sense because if you open up a bag of Starbucks that is, you know, mass roasted,
Yeah.
right? It's over roasted.
Mm-hmm.
And that's why their coffee is terrible.
Yeah.
Only you drink it enough, you acquire a taste, and then it becomes like all things that are mediocre and satisfies the masses, right? But truly, a sufficiently roasted bean is not going to have oils on the outside. And that will be healthier for you because they won't get oxidized because they're protected inside the bean.
Wow.
Okay?
That's cool. Yeah, I had the same thought too. Like, oh, if it looked too dry, I'm like, this can't be good.
No,
Yeah.
but if you look at, you know, like if you go to your smaller batch
Yeah.
roasting companies, you know, like Blue Bottle and the Bay Area and stuff like that, it's dry. Anyway, biodiesel, yes. Here's the mechanism for that. Maybe we'll pick up with the mechanism, and then on Thursday we'll talk about phospholipids. Okay? Cool.